First, I want to start by expressing my appreciation to the organizers, but actually my deepest appreciation to all of you in the audience who are here to listen. It really is greatly appreciated by all of us presenters. Let me give you an introduction to Medeor Therapeutics, where we are changing the organ transplant paradigm. As a bit of a capstone, our focus is personalized and patent-protected cellular immunotherapies. Our focus is exclusively in the U.S. and global organ transplant markets. Our addressable market as of today is greater than 126,000 individuals who undergo a transplant surgery each year. Our concentration is on the kidney and liver transplantation markets because combined they represent 89% of the total. Kidney transplant alone represents 2/3 of the total. These are premium price potential products, and at the moment we have little or no competition in our space. The technology that we have licensed from Stanford University evolved from decades of innovation and competition, and we really appreciate all of the great work which has been done by the scientists. The clinical data that we have from Stanford is highly predictive of pivotal trial success, and that's what we're focused on. The management team of Medeor brings industry, FDA, NIH, and academic expertise. We're currently launching our pivotal phase 3 study under an SPA from the FDA, and based on our horizon of milestones and news flow, are focused on raising our last private round of financing and then heading towards the public markets, market conditions permitting. The problem in all transplants is universal, and that's the fact that all transplant recipients require lifelong immunosuppressant therapy and monitoring of that immunosuppressant therapy. The problem is that all classes of anti-rejection drugs are inadequate and simultaneously highly toxic. Rejection leads to transplant organ loss, which in some settings leads to patient life loss. The toxicity and the cost associated with the therapy also promote drug noncompliance, which just closes the loop and exacerbates the problem. The solution as we see it is on the extreme left-hand side of the slide, and this is a representative diagram of the MDR 10X series of products. Our products are based on proprietary compositions of hematopoietic stem, progenitor, and immune cells, with specific attention paid to modulating the content of T cells in order to ensure engraftment of donor-derived hematopoietic stem cells into the bone marrow of the recipient. If you look in the middle, what you see is our goal here isn't to replace the bone marrow of the recipient, but actually have stem cells from the donor coexist in a state of mixed chimerism with the stem cells of the recipient. It's then cells that are derived from the donor-derived stem cells that lead to the immune quiescence, which is the underlying mechanism of action of our technology. And it's actually antigen-presenting cells derived from the donor stem cells that migrate to lymphoid and thymic tissue and induce clonal deletion. And clonal deletion combined with immune regulation leads to a state of immune quiescence, historically known as immune tolerance. Just to note, and if you look at the bottom of the middle section of the slide, is in animal experimentation, the monitoring of the mixed chimerism often requires a biopsy of a thymic gland or lymph nodes. We're fortunate to be able to have a blood test that we can use in our patients to actually monitor what percentage of circulating white blood cells are of donor origin. The stem cells that we use to manufacture the product and the organ that is transplanted both come from the same individual. That's an essential feature of the underlying technology. The top half of the slide represents the flow of the donor, the bottom half of the recipient, and this represents a situation of a living donor transplant. The donor undergoes GCSF mobilization followed by an apheresis. That product is shipped to our centralized manufacturing facility where the product is manufactured and maintained in a cryopreserved state until needed. The transplant occurs where the harvest occurs, the kidney goes and is implanted into the recipient, and as you might note, nothing is done to the kidney recipient prior to receiving their kidney. There's no conditioning, there is no chemotherapy, there's nothing done. And this was indicated to us in our market research that the surgeons don't want to take patients to the operating room with any kind of drugs coursing through the veins and arteries, or anything having been done to their patient that could compromise the technical success of the surgical procedure. Our conditioning is based on low-dose lymphoid irradiation, and it is commenced following the organ transplantation and demonstration of success of the organ transplantation. Upon completion of the total lymphoid irradiation conditioning, primarily in the outpatient setting, the patient undergoes a single infusion of the MDR-10X product. We have a series of products in development divided by the kidney transplant program and the liver transplant program. The kidney transplant programs, it's different products for different subsets of the kidney transplant market. MDR 101 for the HLA-matched living donor transplant recipient, MDR 102 for the HLA mismatched living donor transplant, MDR 103 for second chance. This is for the patients who say they don't want the product at the time of transplant and then a few months later go, I hate taking immunosuppressants and I want to now get off of them. And then MDR 104 designed for the deceased donor recipient. The first two, MDR 101 and 102, are where we're making the greatest and fastest progress. The MDR 101 pivotal trial, known as the Mercury trial, is launching right now in the US, and the pivotal Venus trial for the HLA mismatch population will launch next year. The same product composition would then move into our liver transplant program. There would be no need, in our opinion, to start from scratch with phase 1 type programs, and we would be able to go right into meaningful mid to late stage clinical programs in recipients of liver transplants. We did very extensive market research, and we used that market research to actually inform our selection of endpoints. And we did this, and I think anyone who went to the plenary session this morning will appreciate that you have to always be thinking forward to what is the market going to want, what is the data that the regulatory agencies will want, but also what is the data that's going to drive adoption of your product. So we took market research drawn from physicians and payers and used it to guide our primary endpoints. What we learned in the market research that really helped us is that the same unmet needs exist in all subsets of kidney transplant recipients, but they're not in the same order. So in the HLA-matched living donor kidney recipient pool, where outcomes are better than in the other groups, still not great, but better, the number one priority was to eliminate all immunosuppressant drugs so that patients do not have the side effects that come from the triple drug immunosuppressant regimens. In HLA mismatched and deceased donor populations, where up to 50% of the kidneys are no longer functional at 5 years after the transplant, the number one goal was give us something that's going to improve the survival of the transplant organ. And that's what we're delivering in our trials. We're basing our trials also on compelling data generated at Stanford University, a phase 2 trial in the HLA-matched population, and a phase 1 trial in the HLA mismatched population. In the HLA-matched population, we had a 96% success rate of achieving mixed chimerism, and 82% of patients were 100% withdrawn from all immunosuppressant drugs. Multi-year durability was demonstrated, and we have up to 10 years follow-up in patients off of all drugs with normal kidney function, normal transplant kidney function, living wonderful lives without the side effects and symptoms associated with steroids, mycophenolate, and calcineurin inhibitors. We have a 100% 5-year death-censored transplant kidney survival and have 0% incidence of any graft versus host disease, new-onset diabetes, BK viremia, and also no grade 3 or grade 4 lymphoid irradiation-associated hematologic toxicities. So highly efficacious, very well tolerated, and this is what's in phase 3 development right now. In the HLA mismatched population, we've identified the optimal CD3 positive T cell dose. We identified that if you had sustained mixed chimerism, these patients developed none of the rejection type episodes that lead to graft failure. Again, excellent 5-year death-censored transplant kidney survival and a side effect profile comparable to that with the HLA-matched population. And that pivotal trial is aimed to begin next year. What do those trials look like? On the left-hand side, the phase 3 trial of MDR 101 in the HLA-matched living donor kidney population is a randomized study. It has an active control arm for safety purposes and a historical control arm for efficacy purposes. It's under an SPA agreement with the FDA. The primary efficacy endpoint is durable freedom from all immunosuppressant drugs. It is a biomarker-driven trial, and as stated already, launching in the U.S. right now. With regards to the MDR-102 Phase 2b study in the HLA mismatched living donor kidney transplant population, again, randomized, active safety and efficacy control arm. The primary efficacy endpoint is comprised of a composite endpoint of events that are known to cause allograft loss. Again, it is also biomarker-driven. Pre-IND submissions have been completed, and the goal is to launch it next year. Both trials are actually open label. What are our near-term milestones at Medeor? Well, first of all, we're getting excited for the first patient enrollment in the Phase 3 study, also submitting the IND for the second study, developing programs and plans and process development related to the deceased donor kidney population and the liver transplant population, raising a Series B financing right now to support all of that work, and focused on pre-IPO preparations. Appreciate your attention and would be happy to field questions either now or in the hallway. Thank you very much.